Greetings everybody. So this is just a tutorial on the heart physiology lab, similar to what I might give you if we were all in class together at the beginning of the lab. So what we're going to be looking at is mainly the Google document lab manual that you all have access to, but also use another page to help us with some additional images. So the main thing about this lab is knowing about the ECG and ways that we can assess cardiovascular, or in this case, more specifically, cardiac health. So we normally do some tests like assessing the activity of the heart with the ECG. We also do some auscultation. We listen to the heart using a stethoscope, and we measure blood pressure in an artery. So those are the three parts of the cardiovascular physiology lab. We're going to start with the ECG, and just remember that with the ECG, what we're looking at here is something that is jumping all over the place, but what we're looking at here is the sum of the electrical activity of the heart. Now, you recently have been studying the change in electrical potential in the different types of cardiac muscle cells, right? So we have the contractile cells, the 99%, the worker cells, and then we have the autorhythmic cells, the 1% that are found along the conduction system of the heart. And they each have some different kind of properties in terms of what they do, and also when we're looking at the electrical activity of them, and we're looking at depolarization and repolarization, etc., they're quite different, okay? But that's when we're looking at the change in membrane potential of an individual cell. And with an EKG, what we're doing is we're measuring the electrical activity of the heart overall, right? And because the different tissues and fluids of the body will transmit a charge, they're conductive, we can measure those changes in electrical activity of the heart as it's working on the surface of the body, right? So that's where we have the electrodes attached to the surface of the body. And what we're doing is much like if we were doing a surgery or a dissection where we make different cuts and we get different views of the organ. In this way, what we're doing is we are assessing the electrical activity along different planes, right? So we may be doing it more horizontally across the atria, across the ventricles, across the right side, across the left side, along the main axis of the heart, which is more of a diagonal. So that's what we're doing when we are using the EKG. Now remember that EKG stands for electrocardiogram. It was originally developed by some scientists in Germany, where K stands for cardia because cardia is spelled with K, but of course here in America it is an ECG because cardio is spelled with a C. So when we look at the typical output from an electrocardiogram, you get this series of three waves that are repeating again and again and again. And there's usually that audio cue, right? The beep, 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 right next to them, right? And if it's in a medical drama, at some point during that show, it's going to go beep, and there's not going to be any waves, right? Because there's no activity in the heart. And then everybody's running around and screaming and crying, etc. Okay, so we're not going to have that, though, today during our lab. So what we're going to be doing is we are going to be assessing what's going on with those three waves. And these are the three waves that we see. So what we have here is a P wave, a Q, R, S wave complex, and a T wave. And each one represents activity in the heart. Now remember, when we talk about the heart contracting, it's not that the whole heart contracts at the same time. The atria are filled and then contract. They push blood down to the ventricles that are relaxed and they're filling, and then the ventricles contract and eject the blood out of the heart while the atria are now filling again in another cardiac cycle, right? So it's atria, ventricles, atria, ventricles, atria, ventricles, like that, okay? Now, because these cells are moving these charged particles across their membrane, it produces electrical activity that, again, we're measuring. So what we see here is the active waves, P wave, QRS wave, and T wave. Now there are these other intervals and segments, but we're not going to worry about that. But it is something that a health professional might assess because these waves should all be present. They should be of kind of a normal, if you will, or average size given your sex and given your age and things like that and your physical fitness. So they should be within a normal range, and there should be a certain amount of distance between them. Not too far, not too short, not extra waves, not missing waves, etc. But in this lab, what we're going to do is we're going to assess these 3 active waves, and we need to know what's going on during them. So during the P wave here is when the atria are getting their signal to depolarize. And with these waves, remember, it's not like depolarization, repolarization like we were seeing before when we were looking at individual cells. Any type of active wave that we see there is basically saying that the heart chambers and those muscle cells that line them are active. It's kind of like a poll or something like that going, hey, what are you guys doing? Hey, there's a bunch of you guys contracting or starting to be active, okay? So the bigger the wave, the more cells are involved, the more activity that's going on. So this P wave right here is atrial depolarization, where the 2 atria are getting their signal to contract. Here, the QRS wave, 2 things are going on. So after the atria are depolarized, then they're going to contract and they're going to eject blood. Afterwards, they need to repolarize and relax and fill again for the next cycle. So that's happening during the QRS wave, that relaxation and filling. So during the QRS wave complex, the atria are repolarizing again. Okay, so atria depolarizing during the P wave, atria repolarizing during the QRS wave. But when the atria are switching over to this relaxation and filling stage, now the ventricles are going into their contraction phase. So the QRS wave is also where the ventricles are depolarizing. Then they're going to contract and eject their blood out the major arteries, and then the T wave is where the ventricles are repolarizing again to be able to relax and fill for the next cardiac cycle. Okay? So that's the first thing you have to know. You have to be able to identify these 3 waves with their letters, and you have to be able to identify what's going on. Atria depolarizing, then atria repolarizing, and ventricles depolarizing, and then ventricles repolarizing. The next thing that we would do is we would actually assess on a QRS wave or a P wave or something like that whether its size is normal or within the normal range. And we can do 2 aspects of it, of any of these active waves. We can do their amplitude or height, and we can do their duration or width. Okay, because if this is normally on graph paper, and when it is, what we see is that on the x-axis would be time, right? These waves are occurring through time, and on the y-axis would be millivolts because it's electrical activity, right? So just like the wall outlet, which is in volts, because that's electric, this also is electric, so it's going to be millivolts, 1/1000th of a volt. Okay, so let's go down then and take a look at what we would do in terms of assessing it. So you're not going to be measuring it directly, but you would be asked to look at the output of an ECG and be able to, again, describe what you're seeing and be able to measure the different waves. So here's some example ones that we see here, right? Repeating waves. And for instance, what we can see here is we can see examples of definite QRS waves. We can see some T waves, and then there's going to be smaller P waves as well. So these are, a little bit different because we were trying to show you some cases where a ECG output might be a little bit odd. Let's switch over to one where it just is an example normal one. So this one, P wave, QRS wave complex, and then T wave that we see there. So there it is represented on graph paper, if you will. And each of these 1 millimeter boxes in the vertical is equal to 0.1 millivolts. Okay? So for instance, if we were measuring the height or the amplitude of this P wave, we would start from where it starts out on the flat, if you will, and then count the number of boxes high it is, right? And this one doesn't quite go a full box, but we'd round it off and say that this is 1 millimeter or 1 box high. And we said that in the vertical, when we're measuring amplitude, each 1 millimeter block is equal to 0.1 millivolts. So the amplitude of that P wave would be 0.1 millivolts. It's very important that you do the conversion and use the units, the correct units, millivolts. If we did the QRS wave complex, we go from the low point to the high point and we count it up. So this isn't starting exactly on a box, and to be sure, on the lab practical, when we're asking you these questions, if you're within 1 millimeter on either side of the actual measurement, or what we think is the actual measurement, you'll be okay. So for this one, we'd start off and there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I would say that this QRS wave is 12 millimeters high. Converting it to millivolts, it's 0.1 millivolts for every 1 millimeter in height. So that would be 1.2 millivolts in terms of amplitude for this QRS wave. And you could probably repeat the same for the T wave, right? So now let's do duration or width. So each 1 millimeter box in the horizontal is equal to 0.4 seconds. So let's take a look at this P wave again. So it starts at about right here, and we see basically two boxes, 2 millimeter boxes in width before it goes back to the flat again. So that means if we say that it's 2 millimeters across, every millimeter is equal to 0.04 seconds. So the duration of this P wave would be 0.08 seconds. Okay? For the QRS wave, you'd go from, again, where it starts to where it finishes. So we'd say, okay, well, that's kind of starting about here. So we'd say 1 and 2 and 3. Sorry about that. So I would say that this QRS wave is about 3 millimeters in width. Each millimeter in width or duration is equal to 0.04 seconds. So this one would be equal to 0.12 seconds. All right? So that's what you need to be able to do. You need to be able to measure amplitude or height and duration of the 3 active waves and use the correct conversion and units. So that's the first major step when we're doing EKG. Well, maybe second, right? Identify the waves, what's going on during the waves, measure the waves. In addition, we have auscultation of the heart sounds. You can feel your heart. We would normally listen to it. You would hear 2 sounds, right? Boom, 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 boom. The first sound is associated with, when we look at it, right? And the atria are contracting, the ventricles contracting. The atria contracting, the ventricles contracting. So what happens is those different valves will close because the blood rushes up against them. They have an umbrella shape and it actually closes them. So when the atria contract, they're doing so from above the atrioventricular valves. It pushes them open and it has the blood drop down to the ventricles. When the ventricles contract, those atrioventricular valves, the blood's going to rush up. It's going to catch like the wind going into an umbrella. It's going to bring those cusps together, close it off. Now the blood is turbulently mixing next to it. It can't go through, so it's got to exit out our other valves, which are going to be like the aortic valve and the pulmonary valve, the semilunar valves, right? So the first sound comes from the AV valves closing, both the left and the right. The second sound comes from when the ventricles are now, they've done their contraction, and they're going into this relaxation and filling stage. What's going to happen is those semilunar valves are going to close. As the blood tries to come back in from those major arteries, those semilunar valves are going to close, and that's going to make a sound as well. So the second heart sound, the DUP sound, is coming from those semilunar valves closing instead. So you need to know what auscultation means, and you need to know the first sound comes from the atrioventricular valves closing. Second sound comes from the semilunar valves closing. All right, blood pressure. We would normally measure blood pressure with the blood pressure cuff. The sphygmomanometer, you should know how to spell that. It's a big fancy word and you should know how to spell it. You get 2 numbers when you measure blood pressure, right? So what you usually get is, who knows, might be 120/70 or something like that. The high number is the systolic pressure, the 120 or the 140 or something like that. The units are millimeters of mercury, because once upon a time these pressure cuffs would have a J-shaped tube with mercury in it, and as the pressure from the arteries is pushing on the cuff, it would push that mercury around the tube. And as that mercury went around the tube, it would go up by millimeters. You would measure how many millimeters it went up, and that would be your measurement for, in this case, systolic pressure. So the units are always going to be millimeters of mercury, abbreviated like that. So systolic pressure is called that because when the ventricles are contracting, they're sending this tidal wave of blood down the arteries, and so the arteries are expanding under this increased pressure, and that's the high point of pressure, so it's called systolic pressure. Now, the ventricles go into a relaxation phase after that where they're filling again. No new blood is coming down the arteries, but after they stretched out, they have this elastic recoil that brings that blood back in and shoots it down the tube, down the artery, downstream. That is the low point, and that's called diastolic pressure, because remember, diastole is when a chamber is relaxing, in this case the ventricles. So diastolic pressure is going to be the low number of the two instead. So systolic pressure, diastolic pressure. Know the names, know what's going on in the heart, specifically the ventricles, during those two. Now, if we give you numbers for systolic and diastolic pressure, you should be able to calculate two things. Number one, you should be able to calculate pulse pressure. We know that the arteries pulse when the ventricles are contracting and relaxing, right? And so the pressure that pushes the walls of the arteries out is called pulse pressure. And the way to measure that would be to measure and take your systolic pressure, subtract your diastolic pressure when they're not pushed out, and that difference is called your pulse pressure. So in order to calculate pulse pressure, it is just systolic minus diastolic. So if we had 140 over 90 before, millimeters of mercury, 140 minus 90 is 50. So pulse pressure would be 50 millimeters of mercury. Mean arterial pressure is a little bit different. Mean arterial pressure, on the one hand, you're like, well, there's two pressures, and if I average them together, then, and divide by 2, right? Then that would give me a mean or an average. But it's not quite like that because the arteries don't spend exactly the same amount of time in systolic versus diastolic. So in order to calculate mean arterial pressure, you take the diastolic pressure, like our 90 from our example, and you add to it the pulse pressure divided by 3. Now that can be a complex number. Like for instance, we had a pulse pressure of 50 divided by 3. You're like, I don't know, 13.3, So in that case, know the actual calculation, diastolic plus pulse pressure divided by 3. And if you can lay it out like that, it's fine. If you want to calculate it or whatever, that's fine. But if you can just lay out the numbers or lay out that type of mathematical formula, then that shows us that you know how to calculate mean arterial pressure as well. Okay, so as you can see, that's the end of our cardiovascular physiology lab. So I hope that helps to prepare you for our up-and-coming lab practical as well. Remember that our lab practical goes all the way back to the blood lab, blood anatomy, identifying the different blood cells, blood physiology when we were talking about things like blood typing, hematocrit, hemoglobin, things like that. From there, we moved into cardiac or heart anatomy, where we had to know all the different layers and membranes and blood vessels of the heart, etc. Then we moved into blood vessels. Remember, with blood vessels, you have to name them specifically. Is it left or right? Name the specific name, subclavian or iliac or something like that. As well, name whether it's an artery or vein. So you got to have all 3 parts to those as you're naming your blood vessels. Don't forget we have microscope slides as well. We've got the blood slide, we've got the cardiac muscle slide, we've got a blood vessel slide where you need to tell the layers of the blood vessels as well as an artery from a vein. And then there's actually 2 slides on there that will be from the lymphatic system or the immune system. We don't really do a lot of lymphatic or immune system in lab. It's more of a lecture thing. But we do have just 2 slides. This slide where it shows a lymphatic vessel, Notice it's in longitudinal section as opposed to cross section like the artery and vein. So all you have to do is identify this as a lymphatic vessel with its V-shaped valve in it. And then we have a lymph node where the lymph, the fluid lymph, kind of percolates and filters through. So you would identify the cortex towards the outside here, more superficial, and then the medulla region, that's more on the inside. There's a connective tissue capsule right on the outside as well. And then these dark circles are little filtration points where we have a bunch of immune system cells that are waiting to do their job and helping to filter the lymph. And these are called germinal centers. Each of these is a germinal center, and they are only found in the cortex. So that's our 5th slide that could show up on our first lab practical. All right. So email me with any questions, and good luck.